Hello everybody, today we are going to talk about gene expression. Gene expression is just the way that we get from our genes to our actual traits. But before we can talk about gene expression, we need to first discuss what a gene is in the first place. So there are two ways to define a gene. You can either say that a gene is part of the DNA that determines a trait, or you can say that it's a segment of DNA that has instructions for a protein. These two definitions are actually the same thing because genes are recipes for proteins and proteins control traits. So you can either say that your DNA determines your trait or your DNA determines your proteins because you are made of proteins. So your trait, which is what your body looks like, how your body reacts to certain things, all of that is because of proteins. The way that we get from DNA to protein is through something called the central dogma. We start with DNA, it turns into RNA, and then that RNA has the instructions to make the proteins. So that means that there are two steps here. One step is going from DNA to RNA, and then the next step is going from RNA to protein. So let's zoom in on that first step. The first step is called transcription. Once again, that's how we get from DNA to RNA. The way that this works is that RNA is complementary to the DNA segment. So if you remember back to DNA replication, the way we're able to make that second strand of DNA is because the nitrogenous bases from one side of the DNA always match up to the complementary bases on the other side of the DNA. RNA works the same way with one caveat. So in RNA, we don't have a T. So if you remember, DNA has four letters, A, T, C, and G. RNA also has four letters, but rather than A, T, C, and G, it has A, U, C, and G. U replaces T, uracil replaces thymine. So this means that whenever we would expect to see a T, we'll instead see a U. So if you ever have to do transcription, there is an easy way to know how you get from your DNA to your RNA. Wherever you see an A, that's going to turn into a U because in RNA, the complementary base to A is U. Wherever you see a C, it's going to turn into a G. Wherever you see a G, it's going to turn into a C. And finally, wherever you see a T, it's going to turn into an A. So if we look at this strand of DNA here, CAT, TAG, GGT, and ACG, this is a strand of DNA. If we wanted to find the RNA strand made from this DNA, then we're going to use the code that I just talked about. So let's look at those first three letters, C, A, T. So we start with the C. We know that C turns into G. Then we know that the A turns into the U, and the T turns into the A. So CAT will become GUA. That's how we go from DNA to RNA. That is the process of transcription. So using that rule, we can make the entire DNA strand into RNA strand, and we end up with GUA, AUC, CCA, and UGC. So now that we've made our RNA, we can go on to the next step, because remember we go from DNA to RNA to protein. So now we're going to look at the next step of gene expression, which is translation, which is how we go from RNA to protein. So one way that you can remember the term translation is by thinking of the way we use translation in normal life. You might translate something from Spanish into English, for instance. It's the same idea here. We're taking something from the language of nucleic acids, DNA and RNA, and turning it into the language of proteins with amino acids. And again, this last step is going from RNA to proteins. So the way that we go from RNA to protein is a little more complex than the way we go from DNA to RNA. And first we need to know how we actually read DNA and RNA. So each set of three letters in DNA and RNA stands for one amino acid. We call these three sets of letters a codon. And if we want to know what each codon stands for, we have to use a codon chart, which looks like this. So at first this can look a little overwhelming to use, but once you get the hang of it, it's actually pretty easy and you can do some translation very quickly. So let's take that same RNA strand that we transcribed a few minutes ago. So again, that is GUA, AUC, CCA, UGC. Notice how each set of three letters is sort of sectioned off from the others. That will make it easier for us to use this codon chart to figure out what it turns into. So again, each 3 letters stands for one amino acid. Those amino acids will all come together and end up creating one protein because remember, the monomer, the building blocks of proteins, are amino acids. So let's do a sample. Let's take GUA, our first codon of our RNA. The first letter of that is G, so we're going to look at the left and look for the row that has G. We can see that that is the row at the bottom. We're only going to look at the row at the bottom. We're ignoring the top 3 rows for now because we know our codon is not in there. Then we look at the second letter, which is a U. For this, we go up to the top and we find the column that is a U. In this case, that is the first column, the one that is most on the left. So we look at where the G and the U intersect and we get that bottom left box. We know that our codon is going to be in there. So we look in that bottom box and we find GUA equals val. Val stands for valine, which is a type of amino acid. So we know that that GUA in the RNA will turn into a val for the protein. Then we just keep using this chart in the exact same way and we can find out what the other 3 amino acids are. I would say pause the video right now to try it for yourself. See if you can get the other 3 amino acids from our RNA sequence. I will let you try that now. All right, did you get valine, isoleucine, ILE, proline, PRO, and cysteine, CYS? If you did, then that's great. You now know how translation works. So you can think of gene expression as a recipe. We start off by having the recipe in our old recipe book. That is the DNA. But we don't want to bring that recipe book around. We don't want to bring it out of the nucleus, so we copy it down, we transcribe it with RNA. Then we take that RNA, this copied down recipe, and we bring it to the ribosomes where it can actually make the food we're talking about. It can make the protein. That's a way to think about gene expression on a broader level, which hopefully may make more sense to you. So sometimes our DNA can actually change, and this is what we call a mutation. What happens is that there's a change in the nucleotide sequence of the DNA, so this means that the letters in the DNA actually change. And this could have an effect on the final protein, which remember, controls our traits. So if you have a mutation, it might change the traits. However, only mutations that happen in your sex cells can be passed on to offspring. If your mutation happens in your eyeball, that won't pass on to your offspring unless that mutation also happened in your sex cells. So there's a few ways that mutations happen. It can happen through dangerous chemicals. It could happen through radiation. It could happen through other environmental sources. Sometimes being out in the sun too long can alter your DNA. And sometimes mistakes just happen when cells are trying to divide. They have to copy DNA, and sometimes they copy things down incorrectly. As for how DNA can change your traits, I would like you to look at this chart here. As you can see, we have the DNA, RNA, and amino acids, just like we saw for the others. So you can see here we started with the DNA TAC, we transcribed that into AUG, and then we translated that AUG into methionine. But I want to specifically look at the second amino acid, the second codon here. We started with GCA, which we transcribed into CGU, and that translates into ARG, which is arginine. However, let's say that a mutation happens, a point mutation happens, which changes the C in GCA into a T. Now that's going to be GTA, and that becomes CAU when we transcribe it, because remember, T turns into A. And then that CAU actually will translate into histidine, HIS, rather than arginine. This will change the eventual protein, which can change the eventual trait as well. And this is why mutations can lead to some interesting different traits, such as having extra fingers, having certain diseases, or just having a different eye color. So that is the grand story of gene expression. Once again, just remember the central dogma. We go from DNA to RNA through transcription. Then we go from RNA to protein through translation. DNA controls our traits because DNA controls our proteins, and proteins make up our traits. If you can remember that and you know how to do the process of transcription and translation, you should be set. I hope that you have a great day and stay safe.